All right. Straight vibing, huh? Okay. Yeah. Yeah. Today we're gonna do. I really don't think it's super hard. I don't think the stuff that we're gonna do is hard, but it's like weird, lots of different weird small things that we got to do before the test. So I don't think it's too bad. Plus, I think I'm really good at explaining this part, which isn't always the case. So, well, you can let me know how it goes. How's that? How's that respiration lab coming along? Parts per million CO2. Yes, love it. I can't see your progress because when I go into pivot, it just shows me, it's assigned to you, but I can't really see. Good job, Noah. You did something nice. I don't remember when I made that due. I probably made it due like end of this week or something. I'm adding a photosynthesis lab today that you're going to be totally on your own. I'm not even going to explain it. I'm just going to throw the pivot like video thing at you and let you mess with it. And you like design your own lab, make your own lab. It lets you, there's only a limited number of things you can do. You can do like bright light, medium light, green light, and then red and blue all together. It's kind of dumb, but I don't know. It's all right to mess with, and you can do a nice little spiel with it. So that's how Carruthers assigns them. She doesn't like give you extra stuff with it or whatever.
She explains nothing. She just goes, here's 3 labs. Have them turned in by Wednesday. Ma'am,
Really?
excuse me. Yes.
I thought I was the crappier teacher. Wow. That makes me feel better about myself now.
Merry Christmas, Volk.
Merry freaking Christmas. Yep. Now I got to delete that from the video. I should just delete the whole video. All right. Okay, so respiration lab, we're good to go. If there's any questions, let me know, whatever. You are, of course, welcome to come to TSS after class today if you do need help with something. I think that the whole photosynthesis lab thing that I'm going to assign to you is pretty straightforward. It's pretty easy. You already are experts on pivot, like you know how to do it and play the video, load the new parameter or whatever. It's pretty simple. All right. Okay, we're going to wrap up photosynthesis. We actually do not have a lot left to do in that chapter, which is super awesome. I was wondering if I could get all this done in 2 weeks, but it looks like it's going to happen. Yeah, our test is this Friday, as announced. It's going to be respiration and photosynthesis, chapters 9 and 10. Wa, I don't know what wa means, but the horse says nay. Or nay as in like no. Okay, anyway, let's take a look at what we talked about last time. Light reactions and Kelvin cycle, just real quickly to get the big punchline. Kind of don't know where my stuff is here. All right, here's my stuff. Oh, there is, I guess, 1 or 2 things I just thought of that I'm going to talk about today. That's kind of hard. It's not that bad, though. All right, big punchline on review sheet. Yeah, there's 2 review things on Google Classroom. There's 2 of them. One is like more like for the multiple choice part of the test. The other one is more like for the free response part of the test. So one is a big batch of free response. I'm hoping to get through a lot of material today, that we have a little bit of time left at the end to look at some of the free response review questions. I'm hoping. We'll see how it goes. Okay, thanks for asking that. Yes. So big punchline of light reactions would be just give me a second here. Oh, I never took attendance. Crap. Oh, geez, attendance. Who's not here? Okay, I got to look at my list. I'm sorry. I forgot attendance because I didn't do a waiting room today because I had to go to the bathroom. So I figured I'd let you guys all in. So now is that going to take me longer? Okay, so just look over light reactions and dark reactions real quick. Kelvin cycle to make sure you're clear on that. So then when I talk about it, we don't have a lot of issues. Let's look at the. So Claire's not here. If you're here and I'm saying your name being not here, just yell, okay? Olivia Craby is not here. And that is all 27. Okay, we're good to go then. All right. By the way, I read those chat notes, like 6 pages I printed out of the chat from last Friday. So you guys are pretty nice. Like you're saying nice stuff. I'm kind of surprised. Very kind of you. So apparently you guys are nice kids. Who knew? Oh, Elena, hers gone. What am I confused about? Elena, are you in here? I counted my numbers wrong. Okay. I thought we had 27 in here. Maybe we have 20. Oh, we have 28 people. That's what. Oh, Claire is here. Where's Claire? Oh, there she. Okay, were you here when I was saying that?
No, I just got here.
Well, okay, so then it's not me. Then it's not me. It's not me, it's you. It sounds like we're breaking up here. All right, okay. Zoom share screen. Why is this so complicated? No, that's not what I want. Pen. Quick summary, light reactions. Light goes in, okay? Water goes in, oxygen comes out. Everybody with me here? ATP, NADPH come out. That's the 2 main products. Oxygen is just a waste product. That's not like the main product of the light reactions. So it's like, hey, what are the light reactions for? Oh, they're to make oxygen. No, that's not the point. The point is to make ATP and NADPH. Okay, those go to the Kelvin cycle where they're going to make your sugars. Okay, so the main product, the Kelvin cycle, is glyceraldehyde 3-phosphate, which is like a mini half a glucose, essentially. You can make glucose out of that. Okay, by 2 of them, you take 2 of them, make a glucose. All right, the other thing that the Kelvin cycle produces is NADP plus and ADP. So that is super important that these feed back into the light-dependent reactions, because light-dependent reactions cannot happen without having these sort of, low energy molecules, kind of like the waste products of the Kelvin cycle. They need that in order to keep going. So if you cut this off, like if for some reason the Kelvin cycle stops, like let's say CO2 is not going into the Kelvin cycle, there's not enough CO2, this is going to stop, and then all of this is going to stop too. Everything's going to stop. So that's a big surprise to some people. They think that these 2 things are very, very separate and that the light reactions can do their own thing and the Kelvin cycle can do its own thing. Nope, they're completely dependent on each other. They have to happen at the same time. NADPH and ATP are necessary for the Kelvin cycle, and then NADP plus and ADP are necessary for the light reactions to do their thing. Or they can't, like if you want to charge a battery, you got to have a discharged battery to put into the charger. A battery charger doesn't like produce battery out of thin air. You have to put the discharged one in there to get a charged one out. What a good analogy. Wait, so how did the LDR start if they needed the product from the Kelvin cycle, and how did they, that is the evolution of photosynthesis. And all of that started with a much simpler process that didn't involve all these components, like the cyclic electron flow thing I talked about last time. And the full explanation to your question, I could attempt now, but it might take me a while, and I probably would fail at explaining it like perfectly, because I cannot tell you all the different steps of the evolution of photosynthesis. But the way that we explain it now is that the parent plant already had the mechanism, gives it to the offspring plant. You know what I mean? So where did the offspring plant get it? Well, it got it from the parent plant. And as the cells grow from the seed, these processes sort of grow together and come about together. All right. And cells come from other cells. So the cells already have, the cells already have an ADP plus in them. They already have ADP or whatever, because cells don't just come from nothing. They come from other cells. So all the like basic molecules were in that first cell in the seed. All right. So that's that. That's the more complex picture. Ideally, you'd have this memorized, but I need you to understand the big picture. You know, the things that come out, the things that go in, the fact that it needs light, the fact that it has an electron transport chain, the fact that there's ATP synthase, there's chemiosmosis, just like with mitochondria, which is pretty sweet. All of those things are important. If you forget like plastocyanin, who cares? Nobody's ever gonna ask you that. Well, I might, but probably not. I already kind of wrote the test. I'm not going to ask you plastocyanin. I'm not going to ask you plastoquinone. But I might ask you about protons getting moved into the thylakoid space. And I might ask you about the migration of protons back through ATP synthase and all of that stuff. Right now I have 20 multiple guesses, and I'll have a couple of free response. I might make this test a little bit shorter this time. We'll see. I haven't decided yet. All right. I haven't really worked it all out in my brain yet, but we'll see. You'll be surprised on Friday. Everybody loves surprises. So it'll be fun. Okay. So Kelvin cycles, same thing, just big picture is really important. CO2 has to come in. Rubisco, I'd like you to know. Rubisco is that super important ancient enzyme. It is, people say it's the most plentiful protein in the world. It is the, by mass, most plentiful protein in the world because any photosynthetic creature has Rubisco in it. Even algae, you know, all the plants do. And Rubisco isn't responsible for grabbing the CO2. It's responsible for carbon fixation, they call this. This process of grabbing the CO2 is called carbon fixation. It fixes the carbons onto Ruby P to make those 3 carbon compounds, 3 PGs, and all of that stuff. And the punchline is here is that we have an extra glyceraldehyde 3-phosphate, an extra 3 carbon molecule left over at the end. That's this guy right here. That is the product of the Kelvin cycle. So if you write the product of the Kelvin cycle is glucose, that's technically not correct. You don't get the glucose till later. That's made later by some other processes where 2 of those are linked together to make a glucose by some other enzymes. So other thing that's important is Kelvin cycle needs ATP. Kelvin cycle needs NADPH, some ATP over here too, in order to make that sugar, that 3 carbon sugar, that glyceraldehyde 3-phosphate. That's difficult to do. It's really hard to take low energy CO2 and shove it together with other carbons to make these 3 carbon sugar, or even to make the glucose. It takes energy to do that, and the energy comes from the light reactions in the form of NADPH and ATP. So overall, really, photosynthesis isn't even that complicated. Yeah, if you go into the details, it's complicated. Yeah, absolutely. But like big picture wise, light reaction, dark reaction interactions, it's not so bad. It's not so bad. All right. Oh, this just is the carbon count. So that's like the same thing, Kelvin cycle, no big deal. This slide just tells you that there's chemiosmosis going on both in mitochondria and in chloroplasts. Both of them have ATP synthase. Both of them, you know, pump protons. Both of them have the protons flow back through ATP synthase and power ATP synthase to make ATP. So both of them have that. So on the test, I put a question that says, discuss, you know, like chemiosmosis or ATP synthase activity in, you know, plant cells and animal cells or whatever. And you got to remember that plant cells do this in chloroplasts and mitochondria. They do it in both. Okay, so animal cells don't have chloroplasts, so they only have this happen in mitochondria. All right. Bacteria do something similar, but they don't use mitochondria because bacteria are like our mitochondria. So they have like their own membrane that they have like an inner membrane on the outside that's inside of the outer membrane where they do take care of this, but we're not going to worry about that's not going to be on my test. Okay. All right. Okay, so now we're going to talk about something completely different. Oh, and by the way, I should show you something. I want to show you that I have some notes for you that I'm not going to go through here, but I want you to know that these are available for you. My typed notes that I normally hand out on paper, you know, if kids are in my classroom, I give them paper handouts of the PowerPoints. I give them paper handouts of the typed notes. This is where they are. Oh, I didn't actually post them. No, maybe I did. Yes, I did post them. Photosynthesis notes handout, that's this. So if you take a look at this, it's, I don't know, it might be a Word. Oh yeah, it's a Microsoft Word. So you'd have to like change it to Google Docs, no big deal. Google does that for you. There's lots of info here. There's some info I didn't talk about, like how do we know, like is the, one of the questions is, what is the source of the oxygen gas that's produced, water or CO2? And well, we obviously know, if you know the light reactions, you know it's water, but this guy did an experiment on it. I'm not going to ask you about that, but it's in these notes. Here's a nice diagram of the Kelvin cycle, beautiful. But then there's a whole bunch of info here. So it gives you all of this, gives you all of this, gives you diagrams. So all that stuff is there. Okay, and I do not have all that in the PowerPoint, but it's here. So this right here, this photorespiration like paragraph, I'm going to talk about now. You should probably write your own notes, but then like I said, you can go back to this and just refer back to it as a resource. Obviously, it's like you don't have time right now to go print this or whatever, but it's there on Google Classroom. Okay. Let me talk to you about photorespiration, because that is a really kind of a misleading name, and kids get confused because the name is so misleading. The name sounds like it's a good thing, like, hey, photorespiration. Photo is light, and respiration is like, hey, it's like, you know, making ATP, like, you know, we use respiration in mitochondria to do ATP. So this is like a great thing. Well, photorespiration is actually terrible for the plant. Let me tell you why. Normal plants, actually, let me get out of this because this isn't what I'm talking about yet. Normal plants, we call them C3 plants. They, C3 plants will, when it's hot and dry out during the day, like on a hot summer day, what they do is they close their stomata. And if they close their stomata, there's a problem. First of all, whatever CO2 is in the space is going to, I might as well put another slide in here. And can I add a slide from here? No. I might as well start drawing some stuff here. So they close their stomata. And what's going to happen is whatever CO2 is in that space in the spongy mesophyll is going to get used up pretty quickly. Okay, so imagine you're a leaf. Here's your stomata, or stoma is just one. Actually, the stomata tend to be on the underside of the leaf, but that doesn't really matter right now. Here's your spongy mesophyll, you know, cells and all that stuff with the air in between. If this gets shut down, here's the palisade mesophyll. What's happening is the CO2 gets absorbed by these cells real quickly. And what you're going to have here is, if these are closed, so if it's hot and dry, stomata are closed, CO2 is going to get really low, or almost down to nothing, basically. And the other thing that's going to happen is all these cells, if they're doing light reactions, are producing oxygen, right? So oxygen is going to go real high. And here's a problem. Rubisco, you know, loves its CO2, and it binds CO2 and fixes it to the, you know, RuBP and all that, and does the Kelvin cycle. It's pretty sweet. But Rubisco is flawed. It's an ancient enzyme. It hasn't changed much over millions and millions of years. It's been kind of like the same enzyme. It doesn't really evolve very quickly. And Rubisco has this flaw where it evolved in an atmosphere where there was a lot of CO2, and now there's a lot less CO2. And the problem is that Rubisco, when there's low CO2 and the CO2 is almost gone, it also binds oxygen. Like it accidentally has an affinity for oxygen also. So if the oxygen is really high and CO2 is really low, Rubisco accidentally will bind oxygen instead. And that's a huge problem because now what's going to happen is Ruby P is going to come in. Rubisco is supposed to grab CO2, but it grabs oxygen instead, which doesn't even have a carbon. It's supposed to make 3PG, a 3-carbon sugar, but instead what it does is it makes a 2-carbon compound. And that 2-carbon compound eventually gets broken down and gets destroyed. So what you're actually doing is if you're binding oxygen to Rubisco, this is really bad, you're destroying Ruby P. And that's not a thing that you're supposed to like eliminate. That's supposed to be a cycle. It's supposed to keep going around in a circle, and you're supposed to keep those components. And the only thing that's supposed to change is you're supposed to feed in 3 CO2 and release 1 3-carbon sugar. And that's not what happens with photorespiration, is that the Ruby P gets destroyed, and you're literally killing parts of your Kelvin cycle that are essentially essential machines in order to keep the Kelvin cycle going. Well, guess what? That's going to cause the plant, if this happens too long, it's going to cause the plant to, that was real tough clue here. I need to hear from you so I know you're with me. To die. That even if it's a super obvious, easy question, if you respond, that lets me know you guys are still with me, right? All right. That causes the plant to die, so that's really bad. So regular old plants like pansies and roses and whatever, like flowers you plant in your garden or whatever, or in your flower pots, those are C3 plants, and they do this. And if you don't water them and it's hot and dry out in the summertime, they can't handle it. They can't tolerate it, and they end up dying because of this, not just because they're drying out. So the drying out is one problem. Not having water going all the way up in the xylem and all that's a problem too, obviously. But the other problem is the stupid photorespiration thing, because Rubisco is flawed and binds oxygen and then destroys parts of the Kelvin cycle, components that we need, like Ruby P. So yeah, that's a dilemma. Plants need their water. Now trees, which most trees, no, all trees that I can think of are C3. They fall into this category, but trees have such deep roots that they can handle it. They can handle it. But there's some kind of plants that have adapted to this and that have evolved in such a way that they can handle dry conditions. Can you think of a plant that can handle dryness much better than most plants?
Succulent.
Cactus.
A what?
Succulent.
Okay.
Cactus.
Instead of being fancy greenhouse talk, just give me like a normal name. Somebody said it.
Cactus.
Your voice said it. Cactus, right? Like a cactus, right? So a cactus is something that we call a cam plant. That's one option. And then another kind of plant that we have is a C4 plant. So cam plants can handle dryness much better. They have a special trick that they use. And then C4 plants can handle dryness much better. That's things like grasses. Grasses, which includes like sugarcane, which includes corn, like a corn plant, and any kind of grass you can think of like on your lawn. Okay, and cam plants are things like cacti and pineapple. I'd give you more examples of cam plants, except your brains are probably full already and I don't want to overwhelm you. Also, I don't know any more examples. Lake Geneva is calling me. I wonder what that's about. Maybe a Nigerian prince wants to give me money. Anyway, okay, so now let's take a closer look at what the strategies are. How do they, basically the punchline here is we want to avoid photorespiration. We need to avoid photorespiration. Call them back. No, you would love for me to be on an embarrassing phone call right in the middle of class. It's probably just spam anyway. So the first approach we're going to talk about is C4. Okay, so these are C4 plants. So they have a special anatomy that's different than regular plants. They have an extra kind of cell. They don't just have the mesophyll cells, and then in the middle of the leaf is the vein, like the vasculature, the tube that transports the sugar to the roots and to other parts of the plant. Like the, you know the veins on the leaf. You know what I'm talking about, the tubing, the pipes, okay? So What C4 plants have is they have an extra kind of cell called a bundle sheath cell. It says it right here, by the way, but it's real small, so I'm going to write it again. Bundle sheath cell. Regular plants, C3 plants, don't have that, but C4 plants do. And what the bundle sheath cell does, it's deeper, further down in the leaf, below the regular mesophyll cells. What that bundle sheath cell does is it shelters the Calvin cycle from oxygen. It hides the Calvin cycle deep in the leaf, close to the vasculature, so there's not so much oxygen from the air exposed to it because it's kind of covered up by those mesophyll cells. That's kind of a nice trick. So it never puts any rubisco in the mesophyll cells. The mesophyll cells they have a new enzyme that it invented. So you would think like, oh, if I'm going to improve the flaw of rubisco, I'm going to make a better rubisco, right? Let's fix rubisco so it doesn't do that thing anymore. Nope. That's not how it happened. That's not how the evolution took place. Rubisco is still the same stupid rubisco, and it still binds oxygen, and it's still flawed. Instead, what these plants did is they invented a whole new enzyme that's called phosphoenol pyruvate carboxylase, which is a name you do not need to know. We're going to call it, for short, Pepsi, the inferior cola, compared to Diet Coke, which is the superior cola, right? Diet Pepsi, blah. Diet Coke, awesome. You can write that down. Those are facts. We're going to call it Pepsi, but it's spelled like this. Pep, phosphoenol pyruvate, C. And by the way, phospho, you know that's phosphate. Pyruvate, you know, is 3 carbons. You know pyruvate. And then carboxylase means it sticks carbon dioxide onto it. Carboxylase means it carboxylases it. It sticks a carbon onto it. That's all. So that's this right here. Pepsi. Okay? So here's what happens. CO2 comes in. It comes into the spongy mesophyll through the stoma. All right? And the CO2, instead of binding rubisco up here, because there is no rubisco up here, it binds Pep-C now. It binds PEP carboxylase. PEP carboxylase combines it with PEP, which is phosphoenol pyruvate, 3-carbon compound, kind of like pyruvate, basically, and it makes oxaloacetate. Oh, well, that's kind of cool. We already know oxaloacetate. We know that from the Krebs cycle, and it's the same oxaloacetate, but it has a different function here. It's used again, but for a totally different purpose. So that's kind of neat. It's the same molecule, new purpose. Oxaloacetate then gets transformed into malate. Well, that's another one we know. Malate we know from the Krebs cycle. Those are both 4-carbon compounds. And all of that happens in the mesophyll cell. Then what happens is the malate loses one of the carbons. It releases one of the carbons as CO2. And it dumps that carbon, that CO2, into the bundle sheath cell, which is further down below where it's sheltered from that evil oxygen. That forms pyruvate. Pyruvate is what's left with 3 carbons, and pyruvate gets converted to PEP, pyruvate with ATP. So this costs a little money. You got to pay for this. This isn't totally free, okay? So you got to pay a little ATP, but it's a small price to pay to save your Calvin cycle. Because now the PEP can go back, and PEP carboxylase binds another CO2, and it keeps doing this again and again and again, and it keeps dumping the CO2 into the bundle sheath cell. Well, what happens in the bundle sheath cell is the following. Rubisco is hiding down here. We know rubisco. Well, if it's rubisco, it's got to be the Calvin cycle. Yep, the normal Calvin cycle, just like always, happens in the bundle sheath cell, and it's able to do that because here it can grab the CO2 without having that oxygen interfering. It's only a little bit of CO2 that's going to make it in there, not high concentrations, obviously. But you don't have that vast amount of oxygen interfering with rubisco and binding to it and then destroying the Calvin cycle. So this is a nice system that these C4 plants use. We call it spatial separation. Spatial means space. We're separating it in space. We're taking the whole Calvin cycle and putting it in a different space to hide it and shelter it and protect it from oxygen way up here. Because up here, there's a lot of oxygen. Down here, there's only a little bit of oxygen. So we're all right. What are we doing on Tuesday if the test is Friday? Party! And by party, I mean the stages of meiosis and how it relates to genetics. That's what I mean by party, which is a party. Because I mean, gamete formation doesn't get any better than that. Wow. Diploid, haploid, oh yeah. I aim to please. It's why I exist. It's to please you people. It's why God put me on this earth. It's to meet your every need. Why would you start a new unit before break? Because I'm out of my mind. I thought I explained this. By the way, do you want to hear good news? You want to hear good news? Today's the 15th, right? Today's the 15th. You know when I gave the respiration photosynthesis test last year with no COVID? I gave it on the 16th of December, which means we're only like a day off or 2 days off. Like we're basically like, now granted, there's some stuff we didn't do. Like we didn't take time to do the labs in class, you know, as much as we did whatever, but okay. Happy birthday. Advanced happy birthday. Anyway. We should totally take the test on Tuesday. No, I don't want to, then we're going to be behind. I was trying to. Oh, that's not why I told you this. You see, you guys twist everything that I say into something. I should just shut up and not tell you stuff. Why do I even say things? I don't know. All of my 3 AP classes are behind. Oh, well, boo-hoo. I don't care about the lesser AP classes. I care about this one. This is the one that matters. The lesser classes, psh. Anyway, can we do Kahoot? Maybe. Let's see if I'm in the mood. Let's see how far we get today. If I'm in a good mood after today, maybe. Maybe. All right. C4. Hey, what do you think about C4? Pretty cool? Good to go? Normally, I would ask my students to memorize this and know all of it, including all the terms or whatever. In this case, it's COVID. So understand the concept, why it's necessary. Because I'll tell you this, a kid that has this memorized inside and out and can't tell me why the plant does this or what good it's for in terms of like photorespiration and all that, that's useless. I'd rather have you not have it memorized and understand the whole photorespiration thing and why this is helpful. The spatial separation part, that would be nice to know. If you forget the name bundle sheath cell, I don't really care. But if you get the concept, that I care about. Answer me. I don't even know what your question is. You guys are so demanding. Jeez. I don't even see a question from you. Okay, wait. So in light-dependent reactions, oxygen is a waste product because the photosystems gain the needed electrons from water. Yeah. That's your question? Yes, the answer is yes. I'm not sure what I don't get the question maybe. I don't know. Oh, okay. All right, there's another one that's less complicated, which is nice, and that's the CAM plants. So I don't even have a separate slide for CAM plants. I just have this half of the slide. So this is all C4 over here. This is CAM. So CAM stands for how do you spell this? Crassulacean acid metabolism. Crassulacean acid metabolism. I'm 87% sure I spelled that right. Nobody calls them crassulacean acid metabolism. Nobody says that. Everybody says CAM plants. Like people that are into biology or botany or plants or whatever, they just say CAM plants because it's a lot easier. Like nobody says adenosine triphosphate. They just say ATP. You know what I mean? So we're just going to call them CAM plants. That's easier. So CAM plants do this. They will at night. They open up their stomata, which is a nice trick. You might say, well, at night, that's stupid because we know the Calvin cycle can't actually take place at night because the light reactions are shut down and there's not NADPH and ATP available. So we can't actually do the dark reactions at night because all that, all those reasons. But they open up their stomata at night anyway, and they take in CO2. And then what they do is they chemically bind the CO2 into these organic acids. They combine the CO2 into these organic acids, which are called the crassulacean acids, 4 carbon acids, which are then stored until daytime. And then in the daytime, they close the stomata. So at night, open stomata. At day, close stomata. And then if you close the stomata, what happens is you don't lose water, because that's the reason they close stomata, is to prevent water loss. You still have this issue of not enough CO2 and oxygen and all of that. However, you can get a bunch of CO2 from the crassulacean acids. So the CAM acids will release their CO2 during the day and give it to the Calvin cycle. So these guys have just one kind of cell. There's just the mesophyll cells. There's not an extra special cell. What they have is temporal separation. Temporal meaning time separation. And they separate the steps of grabbing the carbon and then putting the carbon into sugars between night and day. At night, they grab the carbon, put it into organic acids. During the day, they release the carbon in the form of carbon dioxide, and then the Calvin cycle grabs it. So they got Rubisco and all the normal Calvin cycle stuff going on, and that happens during the day. And if you have enough CO2, because you have it saved up in these acids, photorespiration will not take place as badly, or maybe not at all, because there's enough CO2 there to satisfy Rubisco. It's only when the CO2 is really low, the oxygen's really high, then you get that photorespiration problem. So CAM plants are actually even more effective than C4 plants, because like cacti can survive in deserts. They can survive where there's super low water supply, and they have other strategies too, like the anatomy of the cactus not having leaves, sticking out really far, prevents water loss too, and the big thick sort of structure saves water. Anyway, all right. Any questions on this? All right. So we got C3, normal, C4, grasses, sugarcane, corn, spatial separation, bundle sheath cells. And then we got CAM, cacti, pineapple, temporal separation. All right. This just sums it all up and gives you like a real simple vision of it all. C3, C4, and CAM. And then almost said C5. That wouldn't have made no sense. And that's that. All right. So we're essentially then done with chapter 10. I need to add some extra stuff. I still have 40 minutes, right? So I'm gonna talk to you guys about the labs we should have done, because that pivot lab I assigned you that's kind of a joke. It's super simple. The labs we would have done were way better, like way more interesting. There's two different labs I've done in AP Bio for photosynthesis, never both in the same year, but like one or the other. One is the DPIP lab and the other one is the leaf disc assay. And I'm gonna explain both to you. Why? Because I put questions on the test about them. So the test questions explain the procedure, they explain the process, but they're gonna be super hard if you've never heard of it before. But that's what's gonna happen on the AP test in May. They're gonna like propose like a thing to you that you've never heard of before. You have to read about it, and then you have to figure stuff out, how it relates to the stuff you do know. And that's how this test is gonna be on Friday, but I will tell you a little bit today, so it's not gonna be as bad. So it's gonna be, more reasonable and not so stressed. Devil's smile, yeah, I'm not the devil. I might have sold my soul, but that's a different story. This just sums up what I already told you in the beginning, how the light reactions relate to the Calvin cycle. It might be also good to know that the light reactions take place in the thylakoid membrane. It might be good to know that hydrogen gets pumped into the thylakoid and diffuses back out. It might be good to know that NADPH and ATP are produced in the stroma. Okay, so here they just put this on the bottom and this on top, but these are produced in the stroma, in between those thylakoids in that fluid. And that's where the Calvin cycle is. So it's real close to the Calvin cycle, and it can feed into the Calvin cycle then to get that going. All right. And then the NADP plus and the ADP go back to the thylakoids to basically supply what the light reactions need to do that. This says sucrose. That's BS. It's really glyceraldehyde 3-phosphate. We know that. Oh, it's right here. Okay, so then that tells us later it makes sucrose. Okay, fine. All right, oxygen, we know that part. Obviously, water goes in for that. So water is where the oxygen comes from. CO2 comes in, that's where this G3P. So 3 CO2s come in to make this 1 G3P, or this one here. And also keep in mind that plants can use G3P to build anything. They will use it to build amino acids. They'll use it to build fatty acids. They'll use it to build starch. They'll use it to build cellulose, alpha glucose, beta glucose, fructose, whatever they need to build, they'll use this molecule as your building block, your basic molecule to build up any kind of compound that the plant needs. Because guess what? Plants don't just make sugar. They also make proteins. They also make fats. Okay? So all of that is necessary. Why do we need to add nitrogen fertilizer to the soil? If plants only need water and CO2 to do photosynthesis, which is what we just showed here, why do we have to add fertilizer to the soil, like nitrogen and phosphorus and things like that? Two big components of fertilizer are nitrogen and phosphorus. So like the stuff I dump into my fish tank, this right here, my macros, I don't know if you can read that, but it's nitrogen, potassium nitrate, potassium monophosphate, you know, stuff like that. Tell me why. Phosphorus to catalyze stuff? Yeah, sure, but that's really vague. I want specifically, what do we need? What do we need phosphorus for? What molecules in cells have phosphorus? ADP, or ATP has phosphorus. So to build ATP, you gotta have phosphorus. What's another one that has phosphorus? It rhymes with shmna. DNA has phosphorus. You get the idea. Okay. And what about nitrogen? What molecules have nitrogen? Well, okay, yeah, that's true. But like what really famous group of molecules that everybody knows about, even like a little kid would know about, has nitrogen? Yes, amino acids and proteins. Thank you, thank you, thank you. Very good. So yeah, I mean, NADP has nitrogen, but that nobody knows what that is. You know what I mean? Only you guys know what that is. Anyway, all right. So now let's talk about some of those old labs, because I want you to be familiar with the labs that we didn't do. You know, with COVID, you know, we didn't do a lot of stuff, and I still want you to like know about the stuff that we did. So anyway, one of the labs that we did, the one I've done in the last few years, a real nice lab called the leaf disc assay. An assay is just a kind of test that measures activity of something. Okay, that's just for an experiment. It's a fancy word. The way that works is you take spinach leaves from the store. I literally went to Aldi and got spinach leaves, and I used a hole punch, like a paper punch, and I punched spinach discs. Okay. And we took the spinach discs and we compressed them in a special way that's not important, to get all the air out of the spongy mesophyll, which means that made them sink. So they could sink in water and they sit on the bottom. Otherwise they'd float because they have so much air in the leaf. And then we shined lights on them. And when the light shined on them, what gas would build up in the spongy mesophyll? What gas builds up?
Oxygen.
I don't know what you just said. Can you say it louder?
Oxygen.
Oxygen is correct. So oxygen builds up from photosynthesis, from the light reactions, and it puffs up the leaf discs. It inflates them literally with gas, and it makes them buoyant, and then they rise up. And we can measure how long it takes for them to rise up. The faster they rise up, the more photosynthesis or the less photosynthesis. The faster they rise up, higher rate of photosynthesis or lower rate of photosynthesis? She says higher. Yep, that's right. Okay. So we measure the time, and then, you know, basically the faster they do it, the faster the rate of photosynthesis. Yeah, okay. So that's what that is. So we did the ET50, and ET50 is just simply, where the hell's my pen? Here we go. ET50 is just simply the elapsed time 50%. That means we put 10 discs in there. When 5 of them rise up, we measure that time, and that's my rate then. That's my measurement. That's what I go with. I don't wait for all 10 because maybe one of them died and it's never going to come up. And then if 9 of them come up and one of them doesn't, you get a really skewed measurement. So if we do just 5 out of the 10, then we get a nice, reasonable kind of a measurement that'll work. So that's what we do. So higher ET50, I'm sorry, lower ET50 is a higher photosynthesis rate. Higher ET50 means lower photosynthesis rate. So then you can predict like, okay, red light, effective or not effective? Here, let's just do something. I put white light and I measure my ET50 to be 3 minutes. Okay, now red light. Same like intensity as the white. I just took away the green and the blue and I only gave it the red. What should my number be? Should it be less than 3, or should it be more than 3? Less than 3? Okay, so she says red light is 2 minutes. In other words, plants do better with red light than with white light? Think about that for a second. What's more effective, giving it the push from red and blue, and then, green, not gonna do much, but red and blue together are gonna be stronger or weaker than just red?
Stronger.
Stronger, yeah. So just red is going to be worse than white, which means it won't be 2 minutes. It'll be, just make up a number. Reasonable number. I feel like it would have to be higher than 3. Yeah, so just make up a number. 55? No, I can't try 55. Let's say 5 minutes. Still pretty good. Do we know red is effective with chlorophyll? Is red absorbed? Does chlorophyll absorb red? Can plants use red light? Yeah, absolutely. They love red light. Red light is great. Hey, check this out. Hey, I'll show you. This is totally worth the wasting time here. Totally worth it. Okay, you seeing this here? What color is that? Can you see it or no? Look at what is here. I can't see my screen, so I couldn't tell if you could see it or not. What color was that light mostly?
Red.
Red, yeah, almost all red LEDs. And then the other one has some blue and white and whatever. So I'm actually giving this tank a lot of red because I like to have the red plants look good. Oh, you can't actually see it. So they got some good growth on red there. But anyway, so the point is, red should work pretty well, but not as well as white, because white gives you also blue, and blue is really good. So if I give you blue, I write the word blue in red. That's confusing. Give me a time for blue, just blue now. 7, okay, I'll take it. If you would have said 5, I would have taken that. If you would have said 6, if you would have said 4, if you could take, I don't know. Which is better, red or blue? Probably blue is actually better, but whatever. I'll take 7. 75 SEM. Thank you. So that's fine. Or it could have been 6 or 5 or whatever, but it's got to be greater than 3. It's got to be close. Okay, now I'm just going to give it green, just green. What would I expect to happen to that time? Shorter time or a greater time? Like a lot greater or just a little bit greater? 2 hours, yeah, literally. Okay, we did this experiment. I might have video because the kids actually live streamed it on YouTube, so I might actually be able to find that if I look, but I won't do that now. The kids did that lab last year, and when they shined the green light on those leaf discs, they never came up. They just sat there. I had to tell them, you got to cut it off now. After 15 minutes, I told them 20 minutes, whatever, just shut it down, or you're going to be sitting here till 5 o'clock. It's never going to come up. So the green light didn't work. And the reason why is because chlorophyll does what with green? Does it absorb it and use it?
Reflect it.
It reflects it. It bounces right off. My analogy for that always was this. If you throw donut holes at my face and they go in my mouth and I absorb them, I'm getting energy from those. If you throw the donut hole here and it bounces off and it flies away and it never goes in me, I get no energy from that. That's a good analogy. The donut hole that bounces away is green light. The one that goes in my mouth and I eat it, that's red light or blue light. So now you know, if you ever want to bring me donut holes, they have to be red or blue. They can't be green. See? That's the message we learned from that, the moral of the story. Wait, what? Is everybody Is everybody clear or is there a question? Somebody said, wait, what? Isn't green bad? No, no, it doesn't hurt. It doesn't hurt. It just can't use it. It's like nothing to the plant. It doesn't hurt. There's actually research that says that some green light exposure is good for plants, that there are some pigments that, like minor pigments, that can use it. Because I read a lot of articles about like fish tank plants and like, you know, aquatic plants, and how there's some green LEDs in my LED strip, just a few. And that's supposed to help, but it's very minor. Overall, we can say green is nothing, okay? But it's not bad. So white light has green in it, but it doesn't do any harm. It just doesn't help either. So anyway, all right, cool. So that's the leaf disc assay. That's easier one of the two. And then the harder one of the two is DPIP. I'm not going to tell you what DPIP stands for because I don't want to overwhelm you with fancy terminology. Black light is just UV light. It's not actually black, it's UV. It's on that end of the spectrum past the blue, and it goes into violet, and then ultraviolet is invisible to us, but it's, you know, it can cause problems, and plants can absorb it. Plants can use it. Okay, so the reason I'm not telling you about DPIP is because I don't want to overwhelm you, and because it's complicated, and also because I don't know what it stands for. I forgot. I used to know, but I forgot. And everybody just calls it DPIP, and nobody calls it by the actual name. What you do need to know about DPIP is that it's an analog to NADP+. So it acts just like NADP+. And DPIP in the normal state is dark blue. Normal DPIP is dark blue. Okay, and when it gets, okay, I should start in the beginning here. NADP+, as you know, gets reduced by the light reactions by NADP+ reductase. It gets reduced to NADPH. That's the high energy one. Okay, this is the low energy one. Well, DPIP gets reduced just like NADP+ by the light reactions. The light reactions think this is NADP+ because it's a similar molecule, and it gets reduced to DPIPH. This, we could put a plus here if you want. That one is dark blue. This one is clear, it's colorless. It's completely colorless. So it loses its blue color as it gets energized, and it gets electrons put into it by the light reactions. So we're fooling the light reactions by putting this DPIP in there, and it grabs that instead of NADP+, and it sticks a hydrogen on there, it throws electrons in there, it reduces it, and then it loses its color and it becomes clear. I should really say colorless, but you know what I mean by clear. All right, well, we can measure that with a device called a colorimeter or a spectrophotometer. We use colorimeters in our lab because I borrow those from Carruthers. Carruthers uses those in chemistry for like the Beers reaction or whatever it's called, or Beers law or whatever. So AP Chem people, you know this. So the colorimeter basically is a sensor that can measure how much light passes through something. This thing is called a cuvette. That's just a fancy test tube made out of glass. And then we have a light source over here that can shoot light of different wavelengths and colors and whatever, white light if you want, or blue, or whatever you want, shoot through there. And then this can measure how much of the light gets through. So that's called the percent transmittance. So percent transmittance means how much light gets through. So let's just say there's no photosynthesis at all, and this is really, really dark blue. This is super dark blue. Almost, you know, almost black. Very, very, very dark blue. And no light gets through because no photosynthesis took place. None of it changed to clear. That means we'd have 100% transmittance. That's as high as it can be, or 90 or 95 or something like that. A lot of, oh, I'm sorry. We're talking about transmittance. It's 0%. 0% transmittance. I got it backwards. So 0% transmittance means none of the light gets through. It's too dark. It absorbs all the light that you shine at it, and very little gets through, or none, and the sensor can't sense any. Sorry that I said it backwards. I'm sure that's confusing. I apologize. Are you still with me? Am I losing you? Guys okay? Okay. So if you get 0% transmittance, in other words, none of the light gets through, that means very little or no photosynthesis, a low rate of photosynthesis. That's what that means. Okay, but let's do it again now. I don't have an easy way to erase, so I'm just going to use my eraser tool. Let's do it again. And this time, let's assume there was a lot of photosynthesis. Oh, by the way, the light shining through here, we're not doing photosynthesis. We're not, like this light is just to measure how much took place. The photosynthesis, like reaction, takes place before this. We do that with like suspended chloroplasts and stuff like that. That's not really that important. But let us assume that now I do this again. I'm just going to copy this slide and then make it happen the other way here. I'm sorry, I set this up just right before class, so I'm not super prepared here, but I can make this filled differently. Let's say it's filled like that. Is it okay with you guys if I stay in not presenter view like this? Is it still big enough? Can you see it okay? If I stay in this view, it just won't have the closed caption. Sorry, Amra. Sorry, Amra. Now what happened is we have most of the blue gone. Gain? No, gone. Most of the blue is gone, which means most of that DPIP changed into DPIPH. Most of it was reduced. Okay, so now we're going to have like, I don't know, 90% transmittance. That means most of the light gets through and the sensor can tell most of the light. Very, very little blue left. That tells you high or low rate of photosynthesis. High or low rate of photosynthesis now. High rate of photosynthesis. Thank you. That means a lot of photosynthesis. Okay. So this was a super complex experiment I used to do years ago. It was great. It was a really, really nice lab and really sciencey. Like you got to use the spectrophotometers, colorimeters, and it was very involved. But unfortunately, I mean, we can't do this one now, but I'd still like you to understand the concept. So I've posted 2 video links in Google Classroom for you to watch. It's other teachers explaining both of these methods. One guy is explaining the leaf disc assay. It's like 10 minutes. And another one is a lady explaining the DPIP lab really well. It's from quite a few years ago. And she's standing at her lab table and explaining this and showing you how the colorimeter works and all of that stuff. And she talks through it very, very nicely. So if you're still confused about that, I posted those videos on Google Classroom for you to watch. That's optional, obviously, but you know, if you need it, watch them. If not, then you're good to go. Hey, doing pretty well here. Questions on DPIP and that approach? You really do not need to understand all of the ins and outs of that technique. What you need to understand is that the less blue it gets, the more photosynthesis. And the darker blue it is at the end, the less photosynthesis. So if, okay, so let's say I expose my cuvette ahead of time to green light for 10 minutes. It starts out, you know, as blue or whatever. I expose it to green light, or the technique of that I'm not actually going to get into, but whatever. Let's say I expose my chloroplast to green light, and then I do this test. Would you expect it to be really blue at the end, or would you expect it to be less blue? Green light. So green light, less blue would mean a lot of photosynthesis. So green light, a lot of photosynthesis, right? Everybody agrees? Green light, photosynthesis, yay. Anybody disagree? Stroshan's disagreeing, some other people are disagreeing, they're getting mad. That's the exact opposite of what it is. Green light, no photosynthesis because it's reflected. That's why plants are green. So which means my test tube would be really blue at the end and my transmittance would be really low, would be 0. There's also a way to flip a switch on this machine to make it measure absorbance. That's the opposite of transmittance. So you have to watch out. If the question talks about absorbance, That means how much of the light is taken away, which means if it's really, really blue, the absorbance is 100%. The transmittance is 0%. So that gets a little confusing. On the test, the question I asked you, I talk about transmittance on Friday. So that's why I talked about transmittance here. So it's the same thing as what I talked about now. So it's not backwards. Otherwise, that'd be super confusing. But College Board, you know, if they ask you about it, anything is game, you know, they'll explain it enough, though. If you read carefully enough, you'll be okay. They'll explain it, but they don't expect you to know this ahead of time, but it's nice to have learned about it first, to have heard about it before you get one of these questions. Okie dokie, Smokey. If I shine white light on it, what should I expect my test tube to look like at the end? Really, really blue or clear? White light. Clear, yes, because white light is really good for photosynthesis. A lot of photosynthesis will make the test tube clear, which gives us lots of transmittance, very little absorbance. All right, Hampel, what do you disagree with?
Oh, that was from a while ago.
Sorry, I'm a little slow on that. All right, the last thing we need to talk about is standard error of the mean. This is usually the unit where I teach the kids how to calculate standard error of the mean. I'll go back to my present mode. And many of you, have had, or some of you might have had statistics, and many of you were in my class, so you learned about standard error of the mean already. If you haven't, do not panic. I'm not going to make you learn standard error of the mean calculations before Friday. I'm not going to expect you to do that. I just expect you to be able to interpret graphs that use this as an indicator. So if you know a little bit about statistics, this will be super easy. And if you don't know anything about statistics, this will still be pretty easy. All right, standard error of the mean, just forget about the technicality of it, what it means or anything like that. It's just a way for us to tell if two means or two averages are actually different, significantly, if they're different enough to be real. Like if I say I got this result with red light, let's say, and I got this result with white light. And then I say, oh yeah, white light is more, it's 10 and red light is only 7. This is 7 here and this is 10, let's say. So I'm telling you the 10 is higher. Well, you know something, if my data points were like, for this, let's say for the 7, I had the following data points. I had 4, I had 11, I had 3, I had 0, I had 15, and I averaged it and it came out to 7. Well, those data points are all over the place. They're very unreliable, okay? Which causes my standard or the mean error bars to be really, really big. So now all of a sudden I get these error bars that look like this. We usually do 2 times standard error of the mean in order to get 95% certainty, 2 times, okay? But on the test, I'm not gonna like quiz you on the 1 time versus 2 times or anything like that, but you might see error bars like that, okay? And let's say the same thing with the 10. I got numbers all over the place and the standard error, the mean error bars look really bad. Let's say they look like this. All you have to do is to see if the error bars overlap. Does this overlap with this one? And the answer is yes, this huge overlap. That means that the 7 is not actually different, is not significantly different from the 10. So the 7 versus the 10 are not different enough because those error bars overlap that you can actually say the 10 is higher than the 7. It's not. That was just flukes because your data was so unreliable. Everybody clear? All right, let's look at some more examples of what I'm talking about here. Let's say that I had the following. Let's say I had this experiment here where they're really, really different. You're like, oh yeah, we don't even have to do statistics. I can tell they're different because look how different they look. And this is what the error bars look like. Not really great, okay? Also not really great. And then in this experiment, they were like pretty close, like 8 and 10. They were not very different. You might like, oh yeah, those are not significantly different. I don't even have to do statistics. But then Timmy does the analysis and he gets this. Very, very tight error bars. How do you get really tight error bars? Well, you get that by data like this. Let's say the average is 10 here and your data was like this, 9.5, 10.2, 10.4, 9.8. Are you with me? What do these numbers all kind of look like? They're all really, really close to 10. They're really close to 10. That means this is really good, reliable data. That means if I do if I measure one more time, I'll probably get something really, really close to 10. So those error bars tell me the odds of getting another data point within that realm, within that range of error bars, is 95%. That's what that's telling me. It's pretty good odds. Same thing here. My numbers here were like 7.8, 8.1, 7.9, et cetera, et cetera, et cetera. Really good tight clusters there. Always real close to the 8. And that's how I got that 8 average then, or the mean. So now on a test, I might ask you, okay, which of the two are really different, the 5 and the 10 or the 8 and the 10? And kids are like, oh yeah, the 5 and the 10 are way differenter. No, the error bars overlap. Therefore, these are not significantly different. Here, 8 out of 10, which looks much closer together, the error bars are so tight that they do not overlap. This does not overlap with this one. They're at different levels. There's a space between them. Therefore, the 8 and the 10 is, yes, significantly different with 95% confidence if we do 2 times SEM. Everybody still with me? That was like a super fast statistics lesson. And for those of you that have never had statistics, okay, maybe that was a little confusing, but we already had that question, Sim. How many questions on the test? I said right now there's about 20 multiple guess. I might still change that and there'll be a couple of free response, and I'm probably gonna make the test a little shorter. I already said all this. All right, anyway, hey, that's the end. So we have 12 minutes. I think we should look at some free response questions. Let's take a look at Google Classroom, shall we? Shall. Thanks for asking. Can class just be over? Sure, but not really. Oh, here's the old test. Look at this. This is my test from last year. Look at this. Leaf disk assay, graph slope, DPIP question. Look at that, DPIP. That's last year's test. That CAM and CAM plants and C4 plants. Look at all that, it's beautiful. Little spectrum stuff going on. Action spectrum of chlorophyll. Look at that. Blue and red spike, green is real low. Check that out. This is a nice test. Ooh, the Krebs cycle. I won't put the Krebs cycle memorization questions on the test. You just have to understand big picture. I'm not gonna. Ooh, respiration rate question. Oh, this is actually a SEM question. Check this out. That's a SEM question, because look at this. Big fat error bars, tight error bars. Which one's more reliable? Timmy or Susie? Well, anytime you have a Timmy or Susie question and I ask you like which one did the better experiment, you don't even have to look at the data or anything they did. All you have to do is look at the name. Susie will have it right. Timmy will have it wrong. Feminism, see? There you go. See how politically correct I am? I always make Susie get it right and Timmy is always the idiot. That's how PC I am. I should get like an award. Yeah. Sounds kind of sexist. Yeah, no, it's opposite of sexist. It's the opposite of sexist. If you're mean to boys, then you're a hero. If women are always right and men are always wrong, that's the correct way to say things nowadays. That's how it works. Period. Exactly. All right. What was I saying now? I was going to look at what? Oh, yeah. I was going to look at free response. Check it out. Ooh, that's a nice I wonder if that's going to be on the test. I wonder if Walt just made this picture in Microsoft Paint to put on the test. You get to see all kinds of things here, don't you? There's a test question. All right, Google Classroom. Here we go. What are we doing? Free response practice, bam. All right, this is already, this has been available to you since yesterday, I think I posted this. I don't know when I posted this. So all of these, I would not recommend you doing every single one of these questions. It's going to take you like 12 hours. So don't do that. Look through them and make sure you know the stuff. So I'll take from these topics. I'm not going to say I'm going to use these exact questions, but I'm going to use these topics, okay? And these are fair game. So this is all stuff we learned in photosynthesis and respiration in those chapters. This is all stuff we already know. ATP count, all of that stuff. Thylakoid, electron transport chain, Kelvin cycle, light reactions. Up, here's one. Here's one that maybe we didn't. So let's go F11, make it a little bigger. So here's one that would be a nice big question where you have to read and you have to figure stuff out. So this is a nice free response because you got to read this big paragraph right here. You got to figure out what's going on. You got to predict data, white light, red light, green light, predict the data, make a graph. I'm going to ask you to use Cami to do the graph. So you should know how to use Cami to do a graph. You know what I mean? Like open the thing in Cami and then actually do that. I hope you understand what I'm saying. Okay, so that's that one. So here what happened is the percent transmittance went from 0 to 82 with white. What do you think it should go to with red? 0 to make up a number. 82 is like the best transmittance with white light, the most powerful like photosynthesis action. Now red light, 40. Okay, I'll take anything between 0 and 82. I'm thinking more like maybe 60 or 50, but you want to go 40? What the heck, that's fine. Green light, what will it go to? From 0 to 0, I'll accept it. If you say 10, I'll accept it. If you say 20, I'll accept it. As long as it's less than red. As long as it's less than red. So 15, yeah, 15, but whatever, that's fine. Because maybe the green light isn't pure. Maybe your light source, your LED that you're using or your lamp is not pure green. Maybe it has a little bit of blue and red in there accidentally. That's contamination. And maybe that's causing some photosynthesis. And I told you there's some evidence that shows that green light is effective to some degree. Okay, but if you put 0, that I'll take that. That's fine. That's reasonable because the action spectrum of chlorophyll is basically nothing, no activity at 0 because it reflects it all. So then you fill in the rest of the numbers in and you draw me the graphs. Seems pretty easy, doesn't it? Seems pretty easy. How many people are still going to screw this up? I have a feeling I'm going to put something very similar to this on the test. I haven't done it yet, but I've got a strong feeling. Your other teachers do this? They go through and show you what's going to be on the test? Do they do this for you? See what you got here? I'm going to throw up. Okay, well, don't do that in my room. Anyway, what is this? Oh, this is SEM. So this is SEM. I actually had them calculate SEM. You don't need to do this question. Don't worry about it. But this basically would be, you know, 2 very different bar graphs with tight error bars, and you'd know they'd be significantly different. But you don't need to do that because we didn't actually learn that this year. I will teach you SEM later. We'll go over it later, some other time. CAM plants, C4 plants, action spectrum. Oh, this question is a beauty. This one is awesome. This one was actually on the test in 2013. It was really on the AP test, and it was pretty tough. It's pretty tough. So I would recommend you do this one before Friday. Try to work this one out. It's an actual AP question, just the way it looked on the test, everything exactly the same. Check it out. I thought it was a pretty tough question. It took me a little bit of thinking to figure this out. I had to use my brain for this one. Okay, so check that out. And we can talk about it like maybe, you know, at the end of the day on Friday or on Tuesday. We can go over it. I will not put this one on the test. I will not put this one on the test, but it's kind of fun practice to see what kind of evil stuff the College Board gives us. But overall, pretty nice. A and B are pretty straightforward, pretty easy, and then C is the hard part. C is tough. That's what my kids messed that up back then. Not a lot of them got those points, but overall, not too bad. All right, I'm now done. Maybe I forgot to talk about something. Let me see. Standard error in the mirror for response practice, photosynthesis lab. Oh, I have not talked about the lab I want you to do. So let's take a look at that. So I'm actually going to make it live. So here's 2 videos that are going to explain those 2 labs to you if you want to watch them. One is from Nancy Mancuso and one is from Julian Buss, and they both do a nice job. They're much better teachers than I am, I'm sure, and they do a nice job explaining. Ideally, I would have made that video and showed it to you, but I have no time. I have no time. Here's the lab I want you to do, so I'm going to assign that now. So now it's live on Google Classroom. You can go to that if you'd like. Check it out. We got 2 minutes, right? So we can look at it real quickly. So just go to Pivot, find the colored lights and photosynthesis vulcanized lab. It's already assigned to you. And it looks something like this. So it's basically just this video. There's no questions, there's no introduction, not much instructions here. Kind of tells you a little bit about what I expect. And then you can mess with it. You can change the light color here. You can do medium white, you can do real bright white, you can do green, and you can do red and blue. So I'm a little disappointed. I was hoping Pivot would have more options. It would have been really nice to have an option to take away CO2 here, but they didn't. Can I say one more thing before we leave? Can I tell you one more thing? A common mistake that kids make is that when they a common mistake that kids make is when they look at light-dependent reactions and Kelvin cycle, and you know we have this relationship, you know that in the light-dependent reactions we get the oxygen. Like, let's say we do the leaf disk assay. We're just measuring oxygen production. Let's say we do DPIP. We're just measuring like NADP reductase activity here. Okay, so we're just measuring light reaction stuff. Kids think that CO2 is not important for those experiments. They think that the leaf disk assay will work just fine without CO2. They think that the DPIP assay will work just fine without CO2, that CO2 is only needed for the Kelvin cycle. What they're forgetting is that the light reactions will also stop if the Kelvin cycle stops because of this. You know what I mean? So if I take away CO2 on the leaf disk assay, if I don't give those leaf disks CO2, oxygen production will stop too. The light reactions will stop and the leaves will never come up. So in that leaf disk assay, we add baking soda, sodium bicarbonate. And what the sodium bicarbonate does is it provides CO2 for the spinach discs. And if I don't put the baking soda in there, they won't ever rise because they don't have enough CO2 to make it happen. So that is something I'd like you to keep in mind, that if CO2 is missing, and I kind of wish this lab, this Pivot lab, had the CO2 component, that we could see that it's necessary for these light reactions, you know, to work, but they don't do that. Well, what you're measuring is CO2 absorption. Then basically is how you're watching the CO2 go down. So let's do the white light, the bright one, and let's, you know, as the experiment progresses, the CO2 goes all the way down to 200. So what your rate of photosynthesis measurement is how much CO2 is left, how much you start with and how much is left at the end. So, okay, you have fun with that and do your best with it. I'm giving you a lot of freedom here. Like, you can set it up the way you want. You can do percent change. You can do absolute amount, whatever. You can do bar graphs if you want. You can do rate over time if you want. There's a clock here, I think. I think they give us a clock. Yeah, right here. So you can put a clock in if you want. You can just do bar graphs if you want, up to you. All right, everybody good? Okay, I'll see you Friday for the test, and have a good rest of the week. And thanks for being such nice kids. So I'm lucky to be your teacher. See you later. Hey, Julia, still in here? Oh, Julia. All right, I'll see you later.